Hi, this is Hassan Beg, and I would like to introduce a tool that I have been working on with my supervisor, Jan Madsen. Let's begin with the introduction and motivation. Unlike electronic logic circuits, which are composed of digital logic gates and transistors, genetic logic circuits are composed of gene regulatory networks, which are entirely composed of the biological components of a DNA inside a living cell. The inputs and outputs of an electronic logic circuits are same physical quantity, that is voltage. However, genetic logic circuits are triggered by a combination of different input signals, which could either be any chemical, or a light, or heat, or a combination of different chemical proteins. The genetic circuit then processes these signals to make a logic decision. The logic decision triggers an output, for example, that could be initiation of a transcription process to produce any specific protein. As compared to electronic circuits where signal propagates in separate wires, the signals in genetic logic circuits are molecules drifting in the same cellular volume and hence easily merge with the concentration of other compounds. Therefore, these circuits are very stochastic in nature and are much harder to characterize. The characterization of a device includes the threshold value and a propagation delays of circuit components, like logic gates, which are well characterized for electronic circuits. The field of genetic circuit is not that matured, and thus the area of research for analyzing the threshold value and propagation delay of genetic circuits has to be explored. These experiments on genetic circuits are usually performed in vitro in the wet lab, where biologists are either provided with the ready-made biological models available in the test tubes or are given a specification or recipe from which to prepare it in the lab. Their objective is to analyze the model and verify its functional behavior. This analysis is done, for example, by increasing the molar concentration of input species at any instant of time and observe the effects. It is clearly a very time-consuming process, and due to mishandling of the apparatus or due to any other kind of errors, experimentation needs to be repeated again. Also, the wet lab experimentation for inexperienced users like students or the people who have no biological background might have face expressions similar to this poor guy. In order to avoid repeated and time-consuming in vitro experiments, these biological models are usually validated through in silico analysis by solving reaction kinetics using ordinary differential equations or by performing more realistic analysis through stochastic simulations. According to the best of our knowledge, there exists no in silico tool which gives the user an insight of being in the lab performing live experiments virtually. These facts motivated us to develop a virtual laboratory environment where a user can perform interactive experiments by varying the molar concentrations during runtime and observe the effects on output species without being afraid of overreactions and mishandling of the apparatus. This inspiration led us to develop a software tool named D-VASim, which stands for Dynamic Virtual Analyzer and Simulator. It's a user-friendly environment to simulate and analyze the behavior of genetic circuit models written in SPML file. SPML is a standard way of representing the biological models in a computerized form. So D-VASim takes the SPML file as input and generates a virtual instrument where a user can observe the experiments graphically and vary the molar concentrations through control knobs. The simulation data traces can be used to analyze, verify, and recreate the model's behavior. We have recently added a new functionality of verifying the model by extracting the Boolean logic from the simulation data. Now, D-VASim became a first ever bio tool to facilitate the timing analysis of genetic logic circuits. Please watch the next video for practical demonstration of D-VASim. Thank you.